Bioinformatics.
Okay, welcome to the lab number 4. This is going to be another walkthrough, not intended for you to watch start to finish, but again, just a resource if you want to see how I did things. For that reason, I'm not going to go through all the text and rehash everything that I talked about in lab or that you read and we discussed during lecture, but I will bring it up a little bit where it's important. So this is our 4th lab, so let's go ahead and make a new directory. I'm going to call this lab 4, and we'll cd into that. So we're going to work in kind of a nice confined space. Here I started off kind of with a little bit of kind of write-up of what is an alignment in case you wanted a different way of thinking about it from the lecture, and general ways these things are displayed in a command line format. Bars between matches, dashes between indels, and then dots between mismatches. I show some different examples of alignments and how they can be easy or difficult to pick out which might be the best as far as alignment, but best is a relative term. Again, from lecture, best is relative to whatever scoring system you provide it, and there's many different ways of scoring through scoring matrices for your alignments. In this case, we're going to be using for a lot of the lab a really basic setup where it's 5 points for a match, -4 for a mismatch, -10 for opening an indel, and then -0.5 just for extending the indel. And that's based on our molecular biology knowledge that once an indel is opened, it is a little easier than opening a brand new one. So as you change your scoring alignment, your scoring matrices, so too will the best alignment be changed as well, potentially. So what does a scoring matrix look like when we're actually working with one and not just on a lecture slide? We'll go ahead and I've got a link here so you can download a really popular one, the eDNA full matrix. And here I'm downloading it with curl-big O. You could also use wget. And so we'll download this, and then we can type ls to see that it's in there, and let's take a look at it. You've got a couple options. You could use more, you could use nano and edit it. I like to use cat for stuff like this, just to print it all to the screen, and then we can scroll around. So here, this is very much a matrix, and you're like, whoa, I thought this was supposed to be nucleic acids. Why are there so many? There should only be ATGC. And so what you'll see here is sometimes we don't always have a high certainty of which base pairs were called. Sometimes you know it was, maybe there was an overlap, especially in kind of Sanger sequencing, between maybe an A and a T. And so we'll just use an ambiguity code and call it an A or a T. And so if you haven't heard of ambiguity codes, you can actually click here and see kind of the breakdown of those as well. So we've printed it, we've looked at it. If we're just looking at the unambiguous nucleotides, you can see them here. Again, 5 for a match, -4 for a mismatch. Now, alignment's not the only way to discuss how similar sequences are. You could also think about percent identity, percent similarity. So identity is how many nucleotides are identical across that sequence. Similarity is how many amino acids do they share. So that's kind of more relates to the actual phenotypic impact. And then you can also think of rating these things using e-value, which is almost a statistic for, given the size of your data set, what is the likelihood that you would expect that alignment by chance? And then when we get into more high throughput methods like read mapping, there's also a mapping quality score that we won't get into today. So we're going to do some kind of really simplistic aligning at first, and so to do that, we're going to make use of the emboss package. So we're going to conda install -c bioconda emboss, and you'll have to type yes probably to say that you want that. Yes. And then we're going to install a simple wrapper to this. Now, notice I've got a lot of lines here. We could execute each of these individually. Note that lines that start with this comment sign or hashtag are actually there as comments. Watch, if we were to copy and paste this to the terminal and we execute it, it actually doesn't do anything. So even though this is in the middle of it and it's not a command, we actually, if we wanted to execute all these in a row, we're safe to just copy and paste this whole thing. Typically, the very last line, you still need to hit return. Okay, so we've just done all this stuff, and really what this has done is it has made a new folder called bin within our home directory and made it executable for certain files within there that we just added. Those files we added were this local align and global align files we downloaded. Now, we've also exported the contents of that bin folder to our path. We haven't talked a lot about this, but the path is how your computer knows where to look for details on a command, like ls. ls is in our path. We could actually echo $path, and here are all the folders that when you type ls, that your computer will look for directions on what to say for ls. And so if we type which ls, we'll see that it's actually located within /bin/ls. Now that's a little bit of a tangent, but that's why we just did all this code. So now you should have the ability to use local align as well as global align, which are really simple algorithms for the purposes of just this lab. So if we try to globally align this line versus is aligned, here's what we get. And so we can see that there's some matches, and this based on the scoring matrix that's default to this global align, this would be the highest scoring alignment. So what if we change the scoring? What if we use the argument gap open and change it to a higher penalty for opening gap, or lower the penalty for opening gaps from -10 to 7? Well, now there's fewer gaps or indels, and we're willing to accept more mismatches, basically. There's other things we can do. We can change the value of extending a gap. We can also change what data matrices we want to use for scoring by passing them with the dash data. So if you wanted to use that eDNA full matrix, you could. Okay, so a local alignment is a little bit different. So that tried to align the whole thing, right? So if we want to locally align this line and is aligned, well, now we only aligned 2 letters, NE to NE. Now remember from lecture, that's because you only take the highest scoring islands of overlap, basically, or alignment. And so this is really the only really high scoring section between those 2 sequences of letters. Okay, and so there's plenty of other matrices that we could use, and I've provided links to a bunch here that you could just use wget and download a bunch of right now to your directory. They're just little tables, so they shouldn't take much space. And now let's, just as an example, and you can play around with this and change which ones you want to use. Let's go back to our local alignment and change to Blossom 90. Okay, and now we actually have got a little bit larger of a local alignment because we made the penalties a little less severe with that matrix. And so we were able to get a larger local alignment. Okay, next we talk a little bit about misleading alignments and how particularly when you have long stretches of either repeats or the same base pair, it can be very difficult to identify where the actual differences are. If you have an extra C added within a string of Cs, how does your alignment know whether it was here or here or here? And that can be incredibly tricky. And so this is a lot of just kind of food for thought. But here we run a global alignment, and I'm not going to try and retype all those Cs perfectly. And so look what we get. Rather than picking up that there was just an insertion of 2 Cs into this long stretch of Cs, it actually, because it's such a big penalty to open gaps, it instead would rather force this mismatch than add another C in there. And so we could fix this. If we knew exactly what we needed to do, we could all play around with the gap opening penalty and actually get this to reflect what we know happened in this example, where there were just these 2 insertions of C's that were separate. But we only are able to get to that because we know what our result we want is. This is the problem with reconstructing sequence alignments from things that are diverging over long periods of time, is we don't know what they originally were. And so we have to have a set of parameters that we think are just in general appropriate, but know that they may misrepresent some subset of the data. And the hope is that if you have enough data and you treat it all fairly consistently and well enough, that these kind of instances will get washed out, the bad signal they give will be washed out by the better signals. And so the hope is that examples like this are in the minority of the data that you're looking at. Okay, so let's try something a little bit more genomics-y. And so let's go ahead and grab, just like we worked with last time, some Ebola genomes. And so we're going to get a representative genome from the 1976 outbreak as well as the 2014 outbreak. And so let's ls and see what we have in here. We've got both of our Ebola genomes, and let's just align them with our really simple global aligner and see what happens. So Ebola 1976, remember I'm using tab completion here, and 2014. Now this is going to be a bit longer, this is tens of thousands of base pairs. So rather than have it print all to the screen, let's redirect it and call it Ebola dot align, or let's call it Ebola global dot align. And this will take just a second. And we can go ahead and look at that using, I wouldn't cat it because it's going to be too long, but let's look at the first 40 lines. So let's use head-40 and look at Ebola.global. Okay, and so we can see here's kind of the first 40 lines. We get some output info, what algorithms were run, things like that, the names of the files, what our penalty scoring and matrix were. Here we can see the percent similarity, the percent identity, as well as the percent of gaps, and then the actual numbers here and the overall alignment score. And then you can actually see the pairwise alignment. Here we can see there were some indels at the front, and we'll talk about that later, but do you think these are real indels, or do you think maybe the sequencing probably just started a little bit late, or the quality at the start of this individual here was a little low and it got trimmed off during sequence quality control? So you could really, if you want to see just how much is in even a small viral genome, you could scroll through here and see that we've got about 18,000 bases that are aligned. And you can actually kind of look at this and you can see, oh, there's a mismatch right there. Okay, so that's what the global looks like. Let's look at a local. So you could retype this whole thing, or you could just go back and change the commands. And change the name of the output so you don't overwrite the original. Local. And this will take a second as well. So we can head-40 on the Ebola local alignment and look at this one as well. Now we've got roughly the same identity and similarity. I think the other was 96.8, something along those lines. But here we have no gaps. Now, that's pretty expected if you're doing a local alignment and all the gaps are either at the front or the end. It's only going to keep that high scoring segment in the middle. There's no reason to keep the indels from the front and the back where there's only one stretch from a single sequence there. So this would be a higher scoring alignment because it's leaving off a bunch of stuff that's not comparable. So here we can, think about some of these thought questions. Which of these do you think is more appropriate for this comparison? I would say a local alignment would probably be more appropriate, especially if I were to look at the raw data that led to these genomes being sequenced and assembled. And if we saw that all the indels were due to what originally were base pairs that had been sequenced, but they were really low quality and got trimmed off, then I would say that maybe those indels aren't true indels and that there's just missing data there for one of the sequences. And so local alignment can get around that. I think that kind of answers how there's differences between the global and local alignments as well. You're welcome to go ahead and do this and Google the sequence similarity between chimps and humans or something else that you think are pretty different but still a little similar, and then compare that to the actual divergence between these outbreak strains of Ebola. There are more differences between the outbreak Ebola from 1976 and 2014 than between many things that we think of as being very different species. So viruses, mutate quite frequently, and because they don't have very large genomes, it doesn't take a whole lot of change for it to really shift the percentage as well. Okay, and then here we talk a little bit about from the paper that I shared in the last lab, what the differences they found when they did a much wider comparison of some of these data. And here I ask you to make one more comparison, but not between the 1976 and the 2014, but between two different 2014 accessions. And so to do that, I asked you to kind of go and explore the project page here. And you can go down and go to one of the 249 sequences that they had. And I asked you to get a full one. And so luckily they've identified whether they're full or partial genomes. There's a lot of full ones towards the end. And it really is as simple as going, okay, let's take this one, and we can take this session number here. And all you have to do is pull up what you've done previously and pull that down. So if you didn't remember, we could go back to a little higher up in the lab right here where we e-fetched it. And now we could go use our new accession number and say e-fetch, fetch, database, we want nucleotide. The ID, paste that in there, and format, we want FASTA, and we'll call this Ebola 2014, and we'll call it B, just so we can distinguish it from the other one. Okay, and now what I'd like you to do was do a local or a global or both. I'll just do a local alignment just for simplicity's sake. And we will do the Ebola 2014.FASTA versus the Ebola 2014 B.FASTA. And we'll pipe that to Ebola 2014 local dot alignment. All right, and let's take a look at how similar these things were. So these were both from the same year, but from different regions. And if we look, well, we actually ended up pulling the exact same strain. So they were 100% identical. Actually, excuse me, there were 2 base pair substitutions. Okay, last but not least, I wanted to show you an example of what you would do if you wanted to compare more than a pairwise comparison. So I gave you a very small data set of 6 Turnell regions of the chloroplast from 6 species of a plant. So we could use wget and download that file here, which I accidentally ended with a .text. Again, that's fine. The endings don't really actually matter. So we could use head and look at this. So Cooper. And what format does this look like? This looks like FASTA, right? So what if we were to cat this whole thing? What you're going to see if you scroll through here is this is a FASTA format, and every single sequence is an identifier. On the identifier line is information about the species, where it was collected, what was sequenced. And so here we've got basically 6 different FASTA entries of the different species for a same homologous gene section, basically. Okay, so let's use a multi-sequence aligner. MAFT is a really popular one, so we can install that using conda. So conda install -c bioconda MAFT. And we'll type Y for yes. And we'll use this in really the most basic sense with all the default settings. You could play around with all these things as well, thinking about scoring matrices, points, all that. And we will call the output coop.align. Okay, and so it looks like it was upset, but it's actually just telling you it defaulted to stuff. So here you can scroll up and see what it was actually doing and some of the scores and things like that. But we're not going to get into that. What we're going to do is take a peek at it. So let's cat this coop.align. So this is kind of hard to read. This is still in a format where every single species accession was placed as a different FASTA entry. But now you can see all the indels are represented in here. And so if you were to unroll all these and stack every line on top of each other, it would almost look kind of like a table of sorts. So maybe you can kind of imagine that. But we're not going to look at alignments just like this in this format. What we want to do is do things with them. So maybe we want to do build a phylogenetic tree. And so I'll show you how quick and easy we can build a phylogenetic tree with this really small alignment using a very popular piece of software, RaxML. RaxML can be used for building phylogenetic trees that have hundreds of thousands of entries. It's really scales well to large computing resources and is a really popular phylogenetic tool that uses likelihood approaches that are well suited to nucleotide data, as opposed to kind of maybe what you used in previous classes, more parsimony-based approaches for maybe phenotype. Maybe if you took Bio1 here, looking at building cladograms of beetles and things like that. So I don't want to get into the weeds of every option within RaxML, but if we want to do pretty much the most basic run, we're going to use RaxML HPC -m to specify the molecular evolution model. So we're going to use GTR cat -p. RaxML often wants a random seed, which is basically just a random set of numbers to identify where it can start, basically. Don't read too much into that. -n for what we want to name our output of the analyses, and then -s for our sequence alignment. So many, many more options you could use for this, but we'll just go with the default because we haven't even got to talking about phylogenetics yet. But I would like to give you something to take home from this work that we've been doing. So -s coop.aligned, we hit enter, and it was very, very quick because it's a very short set of sequences. Now, if we type ls, we'll see there's lots of outputs here. What we really want is the contents of this RaxML best tree. You could also look at the parsimony tree. We'll take the best tree. So let's go ahead and cat that to take a look at it. Best tree, hit enter. And this is a tree file. This is basically a set of nested parentheses with both the accession, so what's going to go on the tips of the tree, as well as the branch lengths. And so we can just copy this. Now you could download this to your computer, but this is small enough that we can just copy the text. And you could open it in tree building software to manipulate colors and rotate things and root out groups and all sorts of fun stuff. But for the purposes of today, let's just go to a really basic online tree viewer. I have a link in the lab where you can paste your tree format right here, hit view, and we can see our tree. Now this is a pretty terrible tree. What we have two identical taxa, and then two kind of more divergent, but not divergent enough that we know the order of branching between them. This is currently unrooted, and we could do other things with it, but that is just a very simple example. So if you wanted to try something a little more interesting, what you could do, and I would encourage you to do this, is to go back to the genomes file that we had from Lab 3 and see if you could use MAF to align that. And then, even though it's less than ideal to align whole genomes, align that and then see if you could use RaxML to build a tree. So let's try that. Let's go maft ... to move up a folder to Lab 3 and genomes.fa, and we'll output this as genomes.align. Okay, and this might take a second. So our alignment finished, and again, we weren't using maft in the way that makes it the most happy, but it still gave us an alignment. And for this kind of just first example, this is perfect. So now we can just edit our original RaxML line and now use that genomes.align, give it a new output name, call it genomes, and we should be good to just hit enter. Okay, and so it's going to start to run. It looks like it's a little bit angry, but it's actually going to run all the way through for us. Again, we're doing things very bare minimum right now, but we'll wait. This might take up to 5 minutes to run. Okay, so that should be done. If we type ls now and look at what we have our RaxML best tree for genomes is what we want to look at. So let's cat RaxML best tree for genomes. And this is going to be a lot bigger because there's a lot more things in it. You could open this file up, download it, and open it using CyberDuck or something else. You can also click and drag, whatever kind of is easiest for you to copy that. Go back to our tree viewer, clear this, paste this in here. This is a much bigger tree. And let's go ahead and view this tree. It's going to take a second. But now we actually have, and we can zoom out, because it's actually a decent amount of species accessions if you look at it. And so, these aren't named terribly informatively, but you could think if we had named these by region or time frames, things like that, we might be able to pick out some patterns in the evolution of that virus from a tree like this. So that was just a really kind of nice quick example to kind of show you how this could scale doing things in this method. So with that said, I hope that the lab worked out pretty well for you. As always, reach out if you have any issues. Thanks.